Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing the Telangana State CPGET question paper conducted in the year 2024 for the subject Biotechnology. If you are new to my channel, please do like, share and subscribe. If you want previous year question paper of the same examination being solved, that is of 2023, you can go to my channel Omni Bioentrance. In the playlist C for TSCPGET, you will get it there. So coming to the first question, dash is involved in the synthesis of phospholipids. So it is the smooth endoplasmic reticulum that is involved in the synthesis of phospholipids. Mitochondria, you all know, it is for the energy production. Then cytoplasm, everybody knows what it is. Then endoplasmic reticulum, it is mainly involved in protein synthesis but not in synthesis of phospholipids. It is the smooth endoplasmic reticulum that is involved in the synthesis of phospholipids. Then which of the following cell organelle is present in animal cells but absent in plant cells? So it is the centrosome. It is present in animal cells but absent in plant cells. Nucleus is present in both plant and animal cells. Golgi bodies are also present in both animal and plant cells, whereas chloroplast, it is present only in plant cell. Okay, then centrosomes, what is their role is they help in organizing the microtubules and in formation of cilia flagella, and organization of spindle fibers during cell division in animal cells. Then chromatin is composed of. So chromatin, as you all know, it is made of DNA, histone proteins as well as other non-histone proteins. So here the option is DNA and proteins. But RNA is not a component of chromatin. So answer here is option 2, DNA and proteins. Then in which typical stage of Lamb-Rush chromosome observed? So Lamb-Rush chromosome, they are a type of chromosome that is observed during the meiotic prophase during the oogenesis. So it is typically observed in the diplotin stage of prophase, and it is seen in amphibians and birds. These chromosomes, they are involved in the synthesis of large amount of RNA. So the answer here is meiotic prophase. Okay, what happens in mitotic metaphase? The chromosomes are aligned on the metaphase plate. What happens during mitotic prophase? The chromosome begins to condense. And what happens during mitotic anaphase? The sister chromatids separate and move to the opposite poles. Okay, that's just for your information I told. Then next one. How many hepatocytes would be produced by a series of 4 mitotic divisions that began with the single liver cell? So mitotic division, it will result in the formation of 2 daughter cells. So starting with 1 cell, you will get 2 cells after 1 division. 2 cells will get converted to 4 cells after 2 division. Then you will get 8 cells after 3 division and 16 cells after 4 divisions. So that's the answer. So answer is 16. Then oogenesis. The process that creates female gametes is halted at prophase 1 until puberty. Which of the following describes the DNA content of the female's gamete during her childhood? So oocyte is immature egg and it is halted at prophase 1 of meiosis. Okay. So the chromosome has replicated and it has got 2N content, DNA content. Chromosome has replicated during the S phase. So it has 2N DNA content. So there will be 46 chromosome, or I can say 23 pairs of chromosomes. So total number of chromosome is 46. And each chromosome, they consist of 2 identical sister chromatids. Okay, so 46 into 2 will be 92. So there will be 92 chromatids. So answer here is 46 chromosome and 92 chromatids. Then next one. A gene for corn has 2 alleles. One for yellow kernels and one for white kernels. Cross-pollination of yellow corn and white corn result in ears of corn that have approximately even mix of yellow and white kernels. That is what the question is asking. So that point you have to be careful. Which term best describes the relationship between 2 alleles? So here yellow is there, white is there. They on pollination will give even mix of yellow and white kernels. So there is even mix. There is no specifically that yellow is dominant character. So okay, I will dominate the kernels. No. It is an even mix of yellow and white kernels. So that is a case of codominance. That is when both the alleles have equal effect on the phenotype. That is an example of codominance. Then genetic recombination that happens during meiosis. Then chimerism, that is the presence of 2 or more different cell population in an individual. And incomplete dominance when one allele has partial effect or on the phenotype, but it is not completely masked by the dominant allele. That is incomplete dominance. Then next one. The X-linked recessive trait for color blindness is present in 5% of the males. Okay. If a mother who is carrier and father who is unaffected plan to have 2 children, what is the probability the children will both be male and colorblind? So you have to find out the probability that there are 2 children being born. That is, both of them are sons and both of them are colorblind. What is the probability? So they have crossed a carrier mother with an unaffected father. So this is the carrier mother. So one X chromosome will be having the colorblind gene in it, and father is unaffected. So these are the gametes that are produced. These are the sons and the daughters that they will have. So here it is mainly for male children and they must be colorblind. So the male child becoming colorblind is half, that is 50%. Okay, and the probability that it is a son, that is also 50%. 2 daughters, 2 sons. So 50%. So probability of having a son is 50% and probability of son being colorblind is also 50%. So the probability of having a colorblind son is 50 into 50, that is 25%. Now it is for 2 children. So probability of having 2 children who are both male and colorblind is 25 into 25, that is 6.25%. So your answer here is option 4. Then next one. If the percentage of crossing over between 2 genes is 10, then the distance between 2 genes will be. So it is, the answer should be in centimorgans. So centimorgan, it is the unit of genetic distance where 1 centimorgan corresponds to 1% of frequency of crossing over between 2 genes. So here if the percentage of crossing over between 2 genes is 10 means it is equivalent to 10 centimorgans, because 1 centimorgan is equal to 1% of frequency of crossing over between 2 genes. So the answer here is option 2, that is 10 centimorgan. Then when the activity of one gene is suppressed by the activity of non-allelic gene, then it is known as, that phenomenon is called as epistasis. That is option 3. What is pseudodominance? In pseudodominance, the recessive allele will be expressed. This is because there is deletion or inactivation of the dominant allele. Where hypostasis is the gene has lesser effect on the phenotype than the another gene. And incomplete dominance, I already told you, where the dominant allele will not completely mask the effect of the recessive allele, and it will result in a blend of the phenotype. So answer here is epistasis. So during their experiments, Alfred Hershey and Martha Chase helped confirm that DNA was the hereditary material. Which method did they use this to discover? Transformation of DNA in Streptococcus pneumoniae. It was Griffith experiment in 1928. DNA linkage mapping. It is a technique used to map genes on the chromosome, which confirms DNA as the hereditary material. Radio labeling of DNA and proteins. That is correct, because they labeled the DNA of bacteriophage with radioactive phosphorus and the protein of bacteriophage with radioactive sulfur. Then they allowed the bacteriophages to infect E. coli and found that radioactive phosphorus was transferred to the bacteria, whereas the protein coat, it remained on the outside. So this experiment confirmed that DNA is the genetic material that is transferred to bacteria, while the radioactive protein is not the genetic material. Then X-ray crystallography of DNA molecules, that is for determination of DNA structure by James Watson and Francis Crick. Then in a sample of double-stranded DNA, 30% of nitrogenous base are guanine. What percentage of nitrogenous bases in the sample are adenine? So you know the Chargaff's rule. It states that the percentage of adenine is equal to percentage of thymine, and the percentage of guanine is equal to the percentage of cytosine. So here they have given guanine percentage as 30%. So that means cytosine is also 30%. So total there will be 30 plus 30, 60%. Now out of 100, 60 minus 60 will be 40. 40% will be adenine and thymine together. So adenine alone you have to see. So 40 divided by 2, that is 20%. So the answer here is option 4. Then DNA replication produces a new strand of DNA that is complementary to the parent strand. The parent strand sequence is given. What is the sequence of the complementary DNA? So the answer here is 5' to 3' direction. Okay, so this is the DNA sequence, that is this sequence of the parent strand, 5' to 3'. The complementary to it, we have to write, we'll write it in 3' to 5', but the answer is in 5' to 3'. So you have to write from here 5' C T C G A T C T G C C. So that is there in option 4. So option 4 is the answer. Then what is the composition of nucleotide? So nucleotide, it is having a nitrogenous base, that is adenine, guanine, cytosine, thymine or uracil in the case of RNA. A sugar, it can be a pentose sugar, that is it can be deoxyribose sugar or a ribose sugar if it is RNA, and a phosphate group. So these are the 3 components of nucleotide. Whereas nucleoside, SIDE, it will not have phosphate. It will be having only a base and a sugar. The question is for nucleotide. So a base, a sugar and a phosphate. Then lines, signs, LTRs, DNA belong to. So lines are long terminal repeats and are found in eukaryotes. They are highly repetitive DNA, that is long interspersed nuclear elements. They are lines. Okay, long interspersed nuclear elements, and they are a type of retrotransposon. Signs are SINES. Okay, they have missed the I over here. It is a printing mistake. So it is short interspersed nuclear elements. They are also another type of retrotransposons. Then LTRs are long terminal repeats. They are also repetitive sequence and are found in retrotransposons. In most of the reference that I have taken, it is highly repetitive DNA is more accurate. But in your options, moderately repetitive DNA is also given. I don't know why, but anyway, both may be correct. Okay. Then the DNA polymerase involved in base excision repair is. So it is DNA polymerase beta that is involved in base excision repair. Then DNA polymerase alpha, it is involved in leading strand synthesis during DNA replication. DNA polymerase gamma, it is the mitochondrial DNA polymerase, and sigma, I don't know what it is. I didn't get anything about that. But RNA, you have got RNA polymerase, there is sigma, but I don't know about DNA polymerase. Okay. So DNA polymerase alpha and gamma, I told you. DNA polymerase beta is the one that is involved in base excision repair. Then which DNA repair mechanism relies on the intact complementary strand to guide repair of single strand damage? Okay. So which DNA repair mechanism relies on the intact complementary strand? So there should be a complementary strand and it should there should be single strand damage. So which DNA repair mechanism relies on these two factors? It is more accurate is base excision repair. It relies on intact complementary strand to guide the repair of the single strand damage. Okay. Homologous recombination, it is for double strand break repair, not single strand damage. But your key is having homologous recombination also as correct. Then double strand break repair, non-homologous end joining. It is also a mechanism for repairing double strand break, and it does not rely on the complementary strand to guide the repair. So the answer here is, as per me, more accurate it is base excision repair. Then can you identify the homology and similarity tool from the options below? It is BLAST. Homologous sequence means, homology means sequences that share a common ancestor, and similarity means sequences that share similar structure and function. So it is BLAST that is more can identify homology and similarity tool. Then RASMOL, it is used for visualization of 3D structure. EMBOSS is collection of bioinformatic tools. And PROSPECT is a protein structure prediction tool, not for homology and similarity. So the answer here is BLAST. The first secondary database developed was ProCite, but they have marked the answer as PIR. ProCite was the first secondary database, and it was developed in 1988 by Amos Bayrock. Okay. And PIR, what information I got is, it was initially a primary database and later became a secondary database. So whatever may be the first secondary database, it is ProCite. It may be a printing mistake here in the answer key. Okay. Then which of the following is a protein sequence database? It is PIR. PIR is Protein Information Resource. Then DDBJ is Data Bank of Japan. EMBL is European Molecular Biology Laboratory, and GenBank is for National Center for Biotechnology, NCBI, National Center for Biotechnology Information. and they are primary databases and store nucleotide sequence. Mainly they are involved in nucleotide sequence storage. But they also contain translated protein sequences. But here the question is more specific for protein sequence database. So it is PIR that is more correct. Then which of the following amino acid has a net negative charge at physiological pH? Physiological pH is 7.4. So it is glutamic acid which is having a net negative charge at physiological pH. Histidine will have net positive charge. Lysine also will be net positive charge. Asparagine is neutral at physiological pH because it does not have a charged side chain. Here glutamic acid is having COOH as a side chain. So it will be negatively charged at physiological pH. Then next one. The alpha helix is an example of which of the following structural properties of protein? So alpha helix, it is a secondary structure. Okay. Then primary structure, it is the sequence of amino acids in a protein. Tertiary structure is the 3D shape of the protein and it includes arrangement of secondary structures. Quaternary structure is the arrangement of many polypeptide chains in a protein, just as hemoglobin. It is quaternary structure. Tertiary structure example will be myoglobin. So alpha helix here, it is an example of secondary structure. Okay. How many total molecules of ATP are synthesized from ADP via glycolysis of a single molecule of glucose? So glucose, you know, there is two phases. In one phase there is a utilization of ATP and in the other phase there is synthesis of ATP. So totally there will be a net gain of 2 ATP in glycolysis. But that is not the answer. It is 4 because here there is a step in glycolysis where 2 NADH molecules are produced. So 2 NADH molecules via electron transport chain, they will produce around 2.5 ATP molecules. So approximately 2 plus 2, that is a net gain of 2 ATP in glycolysis plus that ATP produced by NADH will give you 4 ATP molecules. Okay, so the correct answer is 4. If you learn the glycolysis steps, then you will understand what I am telling. First read the glycolysis steps thoroughly, then come to this question. Then, in order to analyze the catalytic effect of two different enzymes on the same chemical reaction. Okay, so the chemical reaction is same, A giving B. Two different enzymes are acting on it. It is best to compare which of the following quantities. The difference in enthalpy between the reactants and the products. The difference between transition state energies. That will be best method because transition state energy is the energy barrier which they must overcome for the reaction to proceed. And enzymes, they do this by lowering the energy barrier, making it easier for reaction to occur. So comparing the difference between transition state energies for the same reaction catalyzed by different enzymes, we will be able to see which enzyme is more effective in lowering the energy barrier and increasing the reaction rate. Okay, hope you understood. So by comparing the difference between transition state energies for the same reaction catalyzed by different enzymes, we can see which enzyme is more effective at lowering the energy barrier and increasing the reaction rate. So the answer here is option 2.